back to that with hypothesis first. Most research questions, when we think about it, are most research questions we always think in clinical trials is the intervention is better than standard of care. Those are usually what's called superiority type. But we have other types of trials, hypotheses, research processes that lead to different types of types and how we design the trials and how we conduct and analyze those trials. So superiority trials is to show that the effect of one treatment on a clinical event or endpoint is superior to another. So these are like placebo trials, and something is another active intervention because I have a new drug that's so much better than the existing standard. So from a statistical standpoint, the way we think about it is we have a hypothesis. All of you are familiar with the null hypothesis and the alternative hypothesis. So the null hypothesis is that we assume that whatever metric we're using to judge whether it is a hazard rate ratio, or it's the relative risk, or it's a difference in means, whatever it is, we use the Greek letter theta, or whatever is that parameter or metric that we are using. Why we use Greek letters? The only reason statisticians use Greek letters is to confuse people, so you hire us. So whatever metric you have, that we assume is the same in both arms. So the differences, or the differences in one group is similar to the other group, or the relative risk in one group and the relative risk in this one. The hazard range one, and the hazard range, whatever it is, that's what is the null hypothesis. The alternative hypothesis is that it's different. Why is it a different one when you say, wait a second, I want the difference here to be bigger. I want it to be smaller. I want to go in one direction. Only, not in both. The reason it is two-sided is actually historically because people cheated. And why people cheated? Because people love significant results. When they designed their studies back in the 1980s, 1970s, in the dark ages of statistical technology. A critical value of Z that was 1.96 or bigger, and they got 1.8, or they were close enough, they would say, wait a second, I'm only interested in one-sided, and one-sided, the critical value is 1.645, so now my 1.8 is significant. So, oh yes, I'm always interested in that. And why? Because people wanted significant results. So what the journals required, and the US FDA in particular required, is no, do always two-sided. If you're interested in one-sided, then do it one-sided at the 0.025. That's it. Don't have to worry about it. So they still use the same critical value. So the test statistics would be, if it's means, it would be t-test. If it's proportion, it's a chi-squared. If it's hazard rate ratio, if it's time to event, we would do those metrics. Very simple analysis. And the p-value would be calculated by the t-test or Fisher's exact test. If you have very large sample sizes and your computer cannot handle Fisher's exact test calculation, then it will give you the chi-square. But the default should be Fisher's exact test. And this is the standard thing. This is superiority trials. Another way of thinking of superiority trials is in terms of confidence intervals. Why? Because I'll talk about non-inferiority in terms of confidence intervals. But if p-value is less than 0.05, then the 95% confidence interval for the difference in the parameters. So now it's the difference in means, or difference of differences of means, or it's in the, when you take the logs, you could have 0, or you could take a relative risk, it's around 1. You include 1 or not include 1. Significant differences when the 95% confidence interval does not contain the difference of 0. Or if it's in relative terms, the value of 1. The same thing. But if it is not significant, then it will contain that null value. Right? Hopefully this is okay with everybody, because this is some standard thing you've seen in your basic statistics course. Okay. This is for superiority. Superiority is the most common and standard way to do things. But sometimes we're not necessarily interested in showing superiority. We want to show this is a new formulation. There is a standard of care for it. I am developing a new formulation, and mine is equivalent to what's already standard of care. Because showing that it's superior, it would have to be so much better. And if something already works, to have something so much better, it's a lot difficult to get. And so I would require a very large sample size if it's just a little bit better. And so I don't want to show that. I want to show it's equivalent. Now, what does equivalent mean? So now we want to show that they're similar to each other. So they're not different by some amount. They cannot be too different. And we want, for example, this is used by, in the pharma industry, looking at generic drugs. They want to say, okay, 2 different formulations of the same active compound are similar pharmacokinetically, pharmacodynamically. They're similar. Or we're doing, for vaccine approvals, they have to show when they're manufacturing vaccines, their lot-to-lot consistency is the same. So they don't want to be different. Or that they're clinically similar. So there's different types of questions that people have for equivalence. We today separate them out, and they say that for the pharmacokinetic and pharmacodynamic characteristics to be equivalent, we're going to call those sorts of equivalent studies bioequivalence. Okay, so they design studies to show that a generic in particular is manufactured by a different company, but it has the same properties as the already approved one. Okay, in India, for example, we have a huge generic medication manufacturing process. It's probably, I think it is the largest in the world here. So every company that develops one of these generics basically is doing these sorts of trials. And these are to show that it's bioequivalent to it. And what do we mean? That the blood concentration levels are on average similar enough. What is similar enough? Okay. So what they're looking at is things like this. They will inject the medication in people with the condition, and they want to see the pharmacokinetics of the medication. So they don't have it. The blood concentration goes up, and then eventually starts going down. Well, they'll look at properties of that medication. How fast does it go up? Which is the top it gets to? How much is concentration is above a certain level that is the clinically important level for having an effect? How long does it stay there? And so they have the length of time, maximum concentration, time to maximum concentration, the area under this. These are properties, we call them pharmacokinetic, pharmacodynamic properties of the medication. This is different from a clinical trial. This is testing the properties of a medication. Then they do it in some people that are randomized to intervention, some are randomized to control, and now they're comparing these properties in there in a small set of people. So yes, there could be a trial, but they're not showing efficacy. They're not showing that one is better or worse than the other. They're showing that they're similar in these properties. They're not clinical condition of the patient. They are characteristics of the drug itself. So we call this now bioequivalence. We have other types of equivalence trials that are clinical equivalence. Now we're looking at the outcome or clinical event that could be a difference in means, or it could be a proportion that do well, proportion that die, or proportion that get better, or we're looking at time to getting better or time to die, and whatever the metric. Again, we use theta, and we say that the difference between the two arms, we want to show that it's less than some value. And less than some value, let's say delta. So we don't want the difference to be greater than some small value. That value may be, of course, in the same units as what we're measuring. So if it's talking about difference in means, that's also the units of the mean. If it's a proportion, then it's difference in proportion, plus or minus 1%, plus or minus 0.5%, a very small amount. Okay. Or the null hypothesis is that it is bigger, so they're not equivalent. And then we do the same test. The problem with this is equivalence then means that the confidence interval, 95% confidence interval, I'll use this, should be between minus delta and delta. So not equivalent is that if it's wider or one of these sides goes over. And it could be that the test one, it's actually better, but it's goes the opposite, goes out in one direction. It turns out to be better. So nobody likes to do these equivalent studies anymore, because even if it's better and your confidence interval goes beyond the better part, maybe if this is the better part, then it becomes a problem. So I'm mentioning this because if you read old papers 25 years ago, people did equivalent studies. We don't do these anymore. This is clinical equivalence. What we do is non-inferiority studies. So non-inferiority studies is I'm interested in showing that my new medication is not inferior to the existing standard of care. If it's better, fantastic. But I'm not trying to show equivalent. I'm trying to show it's not inferior. Okay? So non-inferiority trials like that cryptococcal meningitis that Ananta was talking about, all of those in her platform were non-inferiority to the standard of care. So what is it? The goal here is show the effect of a new treatment is no worse than of an existing treatment. Again, by some amount. How much amount are you willing to tolerate? Oh, it's only 1% worse or 0.5% worse. You don't say, oh, it's 20% worse. No, 20% is too big. So how big can you tolerate? Small amount. Who decides that amount? You. More particularly, not you, your government agency, the Medical Research Council, or Indian Council of Medical Research, or the US FDA. They decide. Because now you are saying this drug is no worse than that. So no, okay, we will approve it if you don't go beyond this. So they give you the boundary. It used to be that people would choose it, but now these regulatory agencies choose it for you. So this is when I have a new investigational treatment. I want to show it non-inferior to a treatment known to be efficacious, or that it's for an approved drug and I have a new drug that is, maybe it's cheaper, maybe it has fewer side effects, maybe I don't have to take it as frequently, maybe only once a week instead of twice a day, and it's more convenient. So but as long as the effect is not inferior. So then we don't want to show that it's superior. We just want to show non-inferiority. Now, it's maybe better safety or ease of administration or less costly. Also, as in the meningitis study, it's to give more treatment options. This is used a lot in cancer because when somebody is on chemotherapy and chemotherapy is not working for this patient, I still may have other alternative chemotherapies that I want to consider that are not inferior for this type of cancer. And I want to give this patient particular choices, more choices to use. Okay. So we use it quite a bit in cancer and infectious diseases. We also use it in things that are established techniques. We do it in cardiology quite a bit as well, that they're trying a new intervention. What was it that they did? They did left atrium, no, left appendage occlusion study. So they were, one of the investigators at McMaster showed that if you have this in the left atrium, we all have this little appendage that affects your atrial fibrillation if you've had a heart attack, and we don't occlude it. So occlude it was to staple it or to keep it from doing things. And he showed that worked. Published a study, fantastic, everybody's doing this. Well, now he says, well, what if I occlude or versus cut? So now we're cutting it. And so I don't want to show that it's superior to the other one, is that it's not inferior to the other one. And so now he's doing non-inferiority study on that. So you don't get a worse result. All right. So the benefit is that since you're going in one direction, if you are lucky enough, very, very lucky enough, that it turns out to be even superior, you can actually test it at the same time. Okay. So we do this, and now people are calling this clinical equivalence, and I don't like that. I prefer non-inferiority. Let's keep it a non-inferiority. So the hypothesis, to make them different, I change this to gamma. But superiority is, let's use this. Superiority is that intervention minus control. The null hypothesis is that there's no difference and that there are differences here. Non-inferiority is in one direction, and saying that it's less than that. Here, equivalence is in both directions. And so we have to get here, we test it against a little delta, the smallest clinically meaningful or important difference. Here, delta, capital delta, is the maximum tolerable difference for non-inferiority. And here, the gamma is the maximum tolerable difference for equivalence. Again, 3 Greek letters, so you can hire us. These are tested one-sided, these are tested two-sided, but this would be equivalent in the critical value. Two-sided would be equivalent to the other. Okay? Let's look at an example. Oops. Oh, I'm blocking you, sorry. I'll stand over here. All right, so these are the hypotheses that we have. What are non-inferiority? It's hard to visualize when you're talking about this. I like to look at it in terms of confidence interval. So I found this good paper that does it. And so it all depends whether your outcome is a good thing or a bad thing. Okay. So if it's a good thing where higher is better, then a superiority is looking at having a confidence interval that does not include 0, and it's on the right side of 0. If it's a bad thing, it's the opposite. So it's the mirror image here. Then you want this to not include 0. You want the confidence interval to be on this side. Equivalence is that you have to be between these two bars, and this is equivalence only. Even if you're better, so here, new therapy is superior, you get it not equivalent. And here, if it's inferior, you get it not equivalent. So That's why we don't do equivalent studies. In a non-inferiority, when the higher is better, you don't want the lower bound of the confidence interval to go to the left of your non-inferiority margin. And unfortunately, these people use little delta for everything. So I will have to fix that in the slides. But non-inferiority for a bad thing, where lower is better, you don't want your upper bound of the confidence interval to be above that. So you focus differently on the upper bound or the lower bound of the confidence interval, depending on whether the outcome is a good outcome or a bad outcome. So why we do this? Because you want to show that it's no worse than that, and for regulatory approval, or from another that is has advantages on it. People call it one-sided clinical equivalence. I don't like it. I'm going to give you an example. So this is for good outcomes. The way we do it, and I'll stand over here so I'm not blocking people. It's if we're doing differences, so now we're differences of means or differences of proportions, then we're comparing. We're comparing to 0. If we're looking at, maybe I'll come over here. I'll get away from everything. If we're looking at ratios, then we're comparing to 1. Okay, so this is for differences on this side. This is for ratios on this side. So for good outcomes, inferior would be if your confidence interval is going beyond the lower bound of the confidence interval is going beyond your minus delta for the non-inferiority margin. Non-inferior would be if the lower bound is above that. And if you're lucky enough that your confidence interval is above 0, you could say, oh, it's better than that. And the same thing, the same image here happens if I'm looking at ratios like relative risk or hazard rate ratios or odds ratios. We would do it this way. The diagram is the same, but just the units are now 1 over here as opposed to and it's 1 over delta and 1 over delta here. For bad outcomes, it's exactly the opposite. So it's now you're looking on the other side and you don't want the, you don't want the upper limit of the confidence interval to go beyond that. You want it to be below that, and if you're lucky enough, you're going to be on this side. So whenever you're reading a paper on non-inferiority, I strongly suggest look at their design and method section and draw this diagram for yourself, because they always present it in a very convoluted way. Doesn't really make much sense until you do the plot. And just plot it, just do a hand plot. They tell you what their non-inferiority margin is. That's that minus, that's a capital delta there. And whether it's capital delta or minus delta for a good outcome, that's what you're looking at to compare. Now, the other thing that I wanted to show here is that the relative size of these things. The little delta for superiority is usually big because you want to have a big effect to show it's better. For non-inferiority, you have a much smaller difference from 0 or from the or the ratio for 1. But the gamma is a whole lot closer for equivalence. And so this will affect sample size. When Professor Jayasilan tomorrow talks about sample size, you will see that the big driver for sample size is that signal. The smaller the signal, the larger the sample size is going to be by a lot. And so that's why equivalence and non-inferiority trials have a much larger sample size than superiority trials. Okay, and I'll show you. Mm-hmm. Right. So it depends there on what is the standard of care. So if you show non-inferiority, that's one thing, but does it become the standard of care is another thing. So let's say I have a new drug. So suppose there is a standard of care in a platform trial now. I mean, normally many people don't do platform trials, but in the case you're doing platform trial like she's doing, and you have a standard of care, okay? So that's an antifungal that works somehow. You have in your platform two new antifungals that you're trying, and you show in the platform that new antifungal one is not inferior to standard of care. You can also show maybe that antifungal two is also not inferior to standard of care, but the government doesn't change the standard of care. Then standard of care stays that. If the government decides, oh, we like the new antifungal two, and we'll make that the standard of care, you still have the platform. Now you have new people coming in. Now your standard of care is the old antifungal two, and you have new people come in with antifungal three or antifungal whatever. Oh, I think what you're talking about is drift. So when you have a drift, that's a different issue. And that is that if you don't want to drift too much away from a good effect, because if you haven't, this is not so much worse, then another one comes, this is not so much worse than this one that was not so much worse now, but it is a lot worse than the original standard of care, that becomes an issue. Yeah. Yeah, that will happen. So that's when the standard of care or the choice of the non-inferiority margin comes into play. What is it? So that's actually what the US FDA has determined. So they set it for you this way, I think. So let's see here. Okay, we already said this. Common pitfall to remind, so showing equivalence. If you test for equivalence and you can conclude significant equivalence, that does not necessarily mean that if you had tested for superiority, you would not have found one. And also, if you test for superiority and do not find, that does not mean that they're equivalents. So they're not interchangeable. That if I don't show this, I show that. If I show this, I don't show that. So that's one thing to worry about. And then, as I said before, statistical significance has no relation with the clinical importance of the result or its meaningfulness. But the non-inferiority margin is an issue. And first of all, you have to select it a priori. You can't change it. Regulatory, if you're designing a non-inferiority trial for something that is a medication or a device or a surgical technique or whatever that is going to be subject to regulatory approval, strongly recommend you have your non-inferiority margin approved prior to designing your study. And it should have clinical decision importance, I guess. So historically, it was done by people, yourself, and you say for a difference of mean, you say a percent of expected mean or percent of the difference. We usually use for binary differences, we use in additive scale, or in multiplicative scale, we use 0.8 and 1.25 for bad outcomes. And that was sort of a standard that was done. This is before the regulatory agencies came into play. And now the US says, no, there is a guidance. You can download it. It's freely available on the internet. If you're doing this, is you want to know the effect of the control or standard before you do your study, and you want the clinical margin to be based on the standard and preserving some of the effect of the standard. how much to preserve becomes an issue. 50%, 75% are the standard things people do. So first, you're supposed to do a meta-analysis of all applicable studies. Then you get what the M1 is, the lower bound of the 95% confidence interval based on that meta-analysis, if you're doing it for good outcomes. And then you set your margin as half of the lower side to preserve of it, or preserve 75%, you do it that way. So an even smaller M2 may be required. That is what you discuss with your regulatory agency, what they force you to do. And so then, so for example, this is a calculation that if from the standard of, from the literature review, you get the minimum of the reference effect is minus 4.5. If you're looking at differences, then requires the test to preserve at least half of it, you would have to have minus 2.25. And if it's want to preserve 75%, you have to have minus 1.125 from the reference. And that would be very, very strict and would require a really large sample size. If I'm going to use this one, this difference becomes very small. The larger the difference is that, but they force you to do that, and your test has to be that. So we will become more clear when we do this for sample size later on. And then the equivalent for large value is bad. It's the opposite. And you'll get this as an example that you can keep. We did this in a study that was just designed recently, looking at, in cardiology, the active control was anticoagulant, rivaroxaban plus aspirin. And they were looking at this test treatment, which was Factorial 11 with aspirin. And the M1 from rivaroxaban studies was minus 20 here. And then they were forced to preserve quite a bit. So then they had M2 was this one here, and they have to use the M2. But this is from many placebo-controlled trials. They were able to get that, and that's how they got from the meta-analysis. They got the M1, and they were forced to do this. So in their particular study, the potential M1 was minus 0.28, and to preserve half of it, they had to the half of the upper limit was around 0.62. And so it's M2 was minus 0.14. So that's half of 0.28. And you could do it in absolute differences, or you could do it in relative differences. And so that was 1.16. And that's what they had to use as a very small margin over there, 1.1. So the tricky statistical issues become an analysis. So the design is one in these non-inferiority trials, but when you're analyzing this, what is conservative behavior? Let's hide here. What is conservative behavior for superiority, like doing intention to treat, is a conservative behavior. That's actually anti-conservative for non-inferiority. And so if you do sloppy work, or there's lots of dropouts and people don't adhere, there's a lot of crossovers, then you would not get a strong effect. And so you would not show much of a difference. So that's why we have to do intention to treat, whether people took it or did not take it. And so in the non-inferiority, it is the preferred way to use the per protocol analysis as opposed to intention to treat. We will discuss this more in other sessions, so I will not discuss it now, but I think in the interest of lunch and starving people, I think we'll take a break here on hypothesis. If there's one or two quick questions, we can take that, or we'll do it after lunch. So save your questions for lunch. chew your question well, and you can digest it and then give us a question after lunch. All right. We'll stop now then. Lunch is upstairs. We have half an hour for lunch. Taskmaster. Yeah, we can. Okay, so there were several questions and concerns about non-inferiority trials. So before we move on, I'll try to explain it maybe slightly differently and see if it's still an issue, we can address it. So non-inferiority trials are ones where we want to show that a new regimen or a new treatment is not inferior to the existing regimen, existing standard of care or control. It depends on whether it's a good outcome or a bad outcome, where we focus our attention. If we're looking at a good outcome, then if I'm taking the difference between the new one and the old one, we would like the difference to be better, right? We want it to be positive. That would be if it's better. But we're willing to tolerate it being slightly worse, not too much worse. So that difference can be negative, but we don't want it to be too negative. So for example, here, if it's too negative, that means that the confidence interval lower bound goes beyond the left side of that margin. And that means it's inferior because it's, in fact, if you look at it, some part of the confidence interval shows it being superior. Some part of the confidence interval shows it not inferior, but there is part that crosses the boundary. And so we say we cannot claim non-inferiority, right? We have to say that it's just like we do not accept the null hypothesis. We fail to reject it. So here we fail to reject the null hypothesis of inferiority, and we cannot say that it's not inferior. Ideally, we could, and that means that the lower bound of the confidence interval is to the right of the non-inferiority margin. So this point here should be above that at the end of the analysis. And then what I said is that some people say, oh yes, in non-inferiority, you get the advantage of testing superiority at the same time. Well, yes and no. Yes, if you are lucky enough that your confidence interval lower bound is above the no difference value, then you can say, yeah, it's better. But to say that it's superior, is it only to be better? Yeah, you could say that. Most people do. Or you may want it to be clinically superior. You would have to be even beyond this part over here. But people are happy with this enough. So, and then if it's a bad outcome, it's the opposite. If it's a, oops, sorry, blocking you. If it's a, if we're looking at differences, we talk about against 0. If we're looking at as ratios, then we're comparing it against 1. But that's just whatever our outcome measure is. So I hope that the design of non-inferiority studies conceptually makes sense, okay? The issue is, how do I choose this minus delta? And as I was saying before lunch, that choice used to be left to the investigator. How much worse are you or that clinical group that you're trying to convince? So the people that work in Uganda in infectious diseases, how much worse would you expect the cure rate to be to still be tolerable cure rate with this antifungal? Oh, 1% worse. 1% is maybe tolerable. 5% may be 2 because that's not good, because the outcome is, or the eventual clinical problem is death. If it doesn't work, death is a problem. So you may not be willing to go 5% worse, up to 5% worse. You may even not even tolerate 1% worse. You may have to say 0.5% worse. It's most we will tolerate. So it used to be that that limit was chosen by the investigator. You were allowed to do it. But regulatory agencies, bureaucrats came into the picture, and they forced things to happen. Most places will copy what the United States Food and Drug Administration does. Why? Because they want their drugs to be sold in the United States. Why? Because they make so much more money in the United States for selling drugs. So every pharma, every government that produces things, like in India, they do generics. They want the generics to be approved for sale in the United States. Why? To make money. Makes total sense. So they would do these non-inferiority studies, and they follow the US FDA guidance. They call it guidance, but it's actually rule. It's not guidance, it's really rule. And the rule is published in this non-inferiority clinical trials to establish effectiveness, guidance for industry. It was prepared for pharma. Okay. And what they say is that the non-inferiority margin is this, should have a clinical importance or clinical meaningfulness. And how do I determine the clinical meaningfulness of a margin? Well, I can base it on my opinion or an opinion of a group of physicians, or they force you to use what has been published in the literature. So they look at the standard of care at that moment, and what is the margin or what is the effect of that standard of care against placebo. And that they call M1. And so you don't want to be, they said you don't have to be that against placebo. You have to be something that is related to that M1, but it's the smallest difference which patients perceive as beneficial and which would mandate, in the absence of side effects and excessive cost, a change in treatment. Okay. And that's what they call the minimal clinically important difference. And that's their M2. And M2 is related to M1 as being smaller than M1. And as I said, you will see tomorrow when we do sample size that sample size is really driven by how small the difference is between the null and the alternative hypothesis, or between the intervention and the control. And here it's going to be very small. And that's why these non-inferiority studies are so large, because the sample size will be bigger. So M2, they give rules on how to calculate M2. You first calculate M1, and then you take 0.5 of it or 0.7, 0.25 of it. So how you determine M1? Based on the literature, usually from a meta-analysis of all applicable studies of the reference versus the placebo. And from these meta-analysis, you get the confidence interval using random study effects. You will see when Professor Tambu talks about meta-analysis, there's a difference between fixed effects and random effects meta-analysis. Random effects meta-analysis give you wider confidence intervals. So it's a penalty that you're paying because they want you to use random study effects. But that'll become clear later. But once you have M1 as the lower bound of that, now you take M2 as based on the M1, either half of it or 75% of it. Or they may even say even smaller. As I said before lunch, if you are contemplating doing a non-inferiority study for a device, for a drug, for anything that is going to be regulated by the Indian Council of Medical Research or any other group that you're working with, I strongly advise you to consult with them and see if the margin you're choosing is one they will accept. They may choose whatever. They may not follow the FDA. They may do more stringent or less stringent. It's up to them. Every country does whatever they want. But as I said, most pharma will follow the US. And so here is actually there's an issue with the slide when it put it on here, it changed the font and it became a little bit bigger. So those minus signs go in front of the. So here was an example. So large values are good. So for a good one, we want the left side of the confidence interval to be the focus of attention. So reference minus placebo from meta-analysis at a confidence interval of 4.5 to 8.5. So we want minus 4.5 as our M1. So minus 4.5 is M1. And what the FDA says, oh, if you want to preserve 50%, it's minus 2.25. If you want to preserve 75%, you want minus 1.125. The smaller it is, the larger the sample size will be. And if large values are bad, then it's the opposite. You'd focus on the 8.5. These examples are given from the guidance. What the guidance has is these numbers. What I did is I drew this because if I read this, I don't understand anything. I need the plot to understand what's happening. So that's why I drew that, and it's not a good drawing, but that's the best I could draw. Yes. Mm-hmm. Yeah. Yeah, we could go anywhere as long as the bottom one is on. Yeah, it can go anywhere. Yeah. Wouldn't that, yeah, if you have a small sample size, you will have, this will be wide, like say, this may be like this, but it is not likely that the effect, see the effect here is here. If you have this wide one and the effect is here, it's actually here. It's not there. You're saying that you're going to get an effect over here. Yeah. You would be if you are lucky enough to get a very large effect. Will be large. If your effect is small, let's say your effect is this here, this point. If you have a small sample size, your confidence interval is likely to be very wide. And not a higher risk, it's almost a definite risk that it will be with a small sample size. If you have a big effect here, then you can afford a small sample size, but you don't know where your effect is. It is because this delta, you choose it very close to zero. You don't expect, see, right now we're doing a trial where it's some bleeding thing. The outcome, safety outcome is bleeding. And they know that with these anticoagulants, to prevent heart problems, they want to prevent strokes. They want to have people not have a stroke. So they're giving them these anticoagulants. But they want to show that this anticoagulant is not inferior to this other one. And they know that the other one causes about 3% bleeding. But that bleeding is not in the brain, it's in non-critical organ areas or whatever. But this other one causes a little bit more bleeding, like 3.2%. So is that 0.2 worse? 3.2, is that too much worse than 3%? No, that's the margin. We have to choose the margin. So we're saying that we already know it's -2. So we expect that we will be somewhere a little bit below here. We don't want it. So we're already below. The effect is likely to be around here. It's not likely to be here. So when I'm around here, I better get a large sample size, because any confidence interval will pass that very easily. And I'll say it's inferior. So depending on what you think your effect is going to be is where you choose the sample size. You're not, if your effect is large and superior already, why are you doing a non-inferiority? Yeah. Right. That's the wrong notion, because they assume that they have a huge positive effect. They're very optimistic. We already know that this causes more bleeding. How do you expect in your study that it will cause a lot less bleeding? You base it on what you know from the literature or previous experience. Yeah, there is a possibility, but sometimes it's a very small possibility. I think it's one of those things like timeshare salesman. They say, oh, this is always fantastic. You will have the best luxury thing if you buy this timeshare. You have that possibility in a lovely thing. Not always true. Sorry.
This is for the organophosphorus poisoning. Pralidoxime, high dose is the recommended drug. So when Dr. A.M. Cherian was head of medicine, did the study. Remember we asked you that is when we realized that this should be approached like, so when you have, he observed that people who are getting, the patients who are getting high dose of P2AM, they're dying more. So therefore he wanted to reduce it to a semi-dose. So the question is, how do you phrase the hypothesis? 1990s, around 2000. So I remember, luckily Kant visited CMC that time. He presented the data to you. And only at that time he said, we phrased the hypothesis. We knew at that time only superiority trials. Equivalence trials were evolving, but that were kept in industries. We did not know the concept of equivalence trial. That is how he has to phrase it like equivalence. Then the non-inferiority came later on, and so on and so forth. Finally, what we have shown is that the low dose was superior to, the placebo was superior to low dose P2A. Is that right, Tambu? That is how we
Was it worse
showed us. Yeah.
than placebo? The placebo? Yeah, so that the
Yeah,
placebo was superior.
superior.
So it was worse than
Placebo was
placebo.
shown to be superior, but the data was, and I think Walter was helping me. Walter, so I used to fax the results to Canada, and because he had the software to analyze the data. So he was taking my manual calculation. So therefore, we finally found out that the placebo was superior. So the low dose was, how do you phrase the hypothesis when you have a low dose P2AM versus placebo? How do you phrase it? So this is about mortality is the outcome.
The more deaths with the drug, with the low dose than placebo.
This is, so this is, so that what we have found was that low dose was inferior, that is placebo was superior. So then the policy now here is they don't use P2A. So I remember there's one mistake which I did. When I say the difference, some people, you just give the exercise tomorrow to these participants, ask them to take the difference. Some people will take the difference as a standard versus the new treatment, standard minus S minus N, new treatment outcome. Some people will take new treatment versus minus the standard treatment, which is correct. Do you understand my question? The difference is minus sign. So it is always, that is a mistake which I did in 2000. I was calculating taking the standard minus new treatment. Walter was taking new treatment minus standard treatments. We were getting a negative, different results altogether. Then he corrected me. Always it is new treatment minus standard treatment. So that is the inferior one which you are hypothesizing minus the standard of care. Then you are correct in this direction of taking the reading.
So here is the test minus the standard.
Yeah, this is, I mean, later on we corrected it, but that is how we need to do. So the question is when you design a trial, I just, you said you should not do it, but the question is, should we go for non-inferiority trial or superiority trial? And so I would go by saying that if after two treatment, someone is inferior, after two, inferior, I would go for non-inferiority trial. It can happen in like a low dose PTAM versus the other one. You can show superiority eventually. There's a possibility that you can show superiority. So therefore, that sometime if the query is whether you phrase the hypothesis as a superiority trial, non-inferiority trial, it is always better to have a non-inferiority trial. Sometimes it helps you to see it as non-inferior. Sometimes it can show you as good as the other one. So you can show equivalence or you can show superiority. Superiority, right? Because like the P2AM, we don't have the direction. So it will reduce the mortality. That is the idea. Low dose will reduce the mortality as compared to placebo because they are not taking any medicine. So, but the reverse has happened. It can happen sometimes. So therefore it is, I mean, the rule is that after two treatment, one is inferior, you just phrase the hypothesis as non-inferior. Is that right? Now, before you start, say for example, we had a study for WHO. We got involved with this WHO. This is about aerosolized form of measles vaccine. So the standard of care is subcutaneous injections. The whole idea is the mothers are not bringing the babies for vaccination because injection. So therefore, this aerosolized form was given. Now, because when you push the vaccine, a lot of vaccine gets wasted. So therefore, obviously that the aerosolized form is inferior. Is that right? After two. This is a gold standard method, that's subcutaneous method. So therefore, you have to phrase the hypothesis as non-inferiority. The question is what is the margin of non-inferiority has to be decided. So do you understand? So if you take any non-inferiority trials, one will be inferior to other one. Sometimes like what we landed up in the PTAM study, that you could get the reverse results. I have that one example. That time I remember the policy was full dose of PTA. So that is what he orders. It was objected in the IRB, but he showed evidences. So his observation was the people who are patients who are taking a high dose of PTA were dying more. That is what his observation. Other group was surviving more. So paying hospital, the patients who could not pay for that drug, I remember it's an imported drug. So therefore they were surviving more. Then from full dose to semi-dose versus placebo. So semi-dose versus full dose, one study. Then after that, semi-dose versus placebo. So both analysis that we get a reverse results. We were showing superiority. So it is possible sometimes.
So anything can happen. But the issue is also you have to sort of plan this in your protocol, right? So you have to specify in your protocol what is your hypothesis that you're going to do. So it's a tricky thing to know. Yes, we think that high dose is going to be superior, and low dose is not necessarily going to be superior, maybe actually inferior. So let's just do a non-inferiority one as opposed to a superiority one for that. So that, I think, is the message here. Okay, so any more questions?